Good morning, everybody. Welcome to day 2 on week 6 of the semester. Remember, this is the week of finals, and we will have our final on Friday and Saturday. Remember that is going to open on Friday and must be submitted by Saturday, which is the end of the semester. Now, we are going to continue talking about the topics of chapter 16. The last thing that we talked about yesterday was the process of denaturation. And when it comes to denaturations, these are different treatments that can happen to proteins in which we are going to be affecting specifically the secondary, tertiary, or quaternary structure. Understand that if something affects secondary, then is going to affect the one that goes after. That's why we can see that, for example, the first treatment, which is the heating above 50 degrees Celsius, or agitation, since it affects secondary, will affect tertiary, quaternary, structural level of a protein if it's available. If it's not available for that protein, then it won't be affected. Now, when it comes to this concept, remember that denaturation, pretty much what it does is that it unfolds the particular protein. So now we're talking about proteins that don't have their original conformation. They are unfolded, and depending on the purpose of that protein in that organisms, then this can affect how the protein actually is active or not. Now, today, we are going to move on and talk about specifically a class of proteins that are very important in biochemistry, and those are enzymes. As you can see the definition here, an enzyme is going to be a protein that acts as a biological catalyst, okay? So if we remind ourselves what a catalyst is, this is going to be a molecule that is going to increase the rate of the reaction. Let me just write that as a note. So remember that a catalyst increases the rate of a reaction. My shorthand for reaction is RXN. And remember that this is done by lowering the activation energy for the reaction, okay? Now, when we think about enzymes, again, these are proteins. They're made of a combination of those 20 amino acids that we discussed in class yesterday. When we look at the overall structure of a protein, which we can see in the figure in the bottom of the slide, this picture is representing what when we look at an enzyme, how does it really look like? So when it comes to the parts of the enzyme, the most important compartment in an enzyme is going to be the active site. And the active site is where the chemistry happens. So whatever chemical reaction that the enzyme is involved in, all of the reaction is going to happen in the active site. Now, understand that in biochemistry, we don't call things reactants, okay? In biochemistry, a reactant is also called a substrate. So reactant is the same thing as saying substrate in biochemistry. As you can see, the word products is still utilized. So if a substrate is going to interact with an enzyme to produce a product, you can see that the binding of that substrate happens at the active site. And then we can see here when we zoom in onto what is happening specifically to the substrate, we can see that the amino acids that are sitting in that active site are going to have interactions with the substrates in order to produce product, okay? So all of this is mediated again, by functional groups, which is what we spent talking at least the last week and a half of the semester. Everything has to do with those functional groups from organic chemistry and how they're going to proceed to make this reaction happen. So as I mentioned, enzymes are biological catalysts, and understand that in the body, nearly all chemical reactions are done by enzymes. So we need enzymes, okay? As I discussed a few moments ago, we are going to be utilizing enzymes because it increases the rate of the reaction, because it lowers the activation energy for the reaction. So here we have the following example. In the body, as we discussed in chapter 10, we have the ability to convert CO2 and water to carbonic acid. If you guys remember, this is how the blood buffer system actually is created, okay? So CO2 and water, using an enzyme, is converted to carbonic acid. Then this carbonic acid is going to then create protons and hydrogen carbonate or bicarbonate. Now, if this reaction is done without the enzyme, then the blue trace line that we have in the energy diagram is the one that tracks the potential energy of the different compounds that are going to be formed. The line that is in lavender, it actually traces the potential energy of the reaction progress. And we can see that in the presence of that enzyme, the potential energy of the reactants and the products is not changed. The only thing that it is modified once we have an enzyme in it, because this is the one with enzyme, and that's the one without an enzyme. The only thing that we are modifying in this system with the presence of the enzyme is that now the activation energy is lower. So we don't have to climb such a large hill. Now the hill is going to be much smaller, and you still get to your product. Now, when it comes to enzymes and their names, understand that their names are varied, okay? One of the most common things that we see in biochemistry when it comes to enzymes is that many names end with the suffix "ase." So whenever we have an enzyme and you have certain names in front of you, the one that ends in ASE is going to be the one that is an enzyme, okay? Sometimes the name of the enzyme even identify the substrate that it has. So for example, sucrase, which is the enzyme that takes sucrose and it breaks it down to glucose and fructose, it has the name of its substrates. Other names are going to just tell us what type of reaction the enzyme actually does. And as you can see, if an enzyme is named as an oxidase, then it catalyzes an oxidation reaction. Now, there are common names for enzymes, and those common names are just assigned by the people that discover them, but it doesn't give us a specific idea of what the enzyme does. You just basically memorize what they do. So for example, pepsin and trypsin, which are enzymes that are involved in the hydrolysis of amide bonds in proteins, you just have to know that that's what they do, because the name doesn't give us an idea of like, oh, this is the specific reaction that they catalyze. Now, it is very important to note that because these enzymes are going to be involved in catalysis, in other words, it is going to be behaving as a catalyst in a reaction, there are a specific classification for the types of enzymes based on the reactions that they actually catalyze. And there are 6 main classes, okay? For those of you that are going to take physiology, once you start studying the way that metabolism works and how the body utilizes biomolecules in order to derive energy, you will see the action of these enzymes into those pathways to see how the body creates that ATP molecule, which is how we store chemical energy to derive energy for the cells to function. But in general, this is the classifications of the enzymes. So if your enzyme is known as an oxidoreductase, the reaction that is going to catalyze is going to be an oxidation reduction reaction. If your enzyme is specifically a transferase, is going to transfer groups of atoms. It could be oxygens, it could be sulfurs, it could be nitrogen, it depends. Basically, when you are trying to define these, you have to look at the substrate and the product to see what is the change in the functional groups in order to define the different classes. When it comes to a hydrolase, these enzymes are going to utilize water to hydrolyze a particular substrate. Lyases are going to either add or remove atoms, but we know that they are different from transferases, especially the lyases that add atoms, because lyases tend to form double bonds or a new ring as the product of the reaction. We also have enzymes that are known as isomerases. And whenever you have an isomerase enzyme, understand that what's happening is that we are rearranging the atoms from reactant to product. Now, understand that whenever these isomerases are acting on the substrate to create a product, what we have is mostly constitutional isomers. In this class, we don't observe different examples for the enzyme classes. We basically just discuss the definitions for them. Lastly, we have ligases. And when it comes to ligases, understand that they're going to bring two small molecules and is going to bind them together. Now, when it comes to enzymes, ligases particularly are going to utilize that important energy molecule called the ATP in order to be able to bind the small molecules into one product. So if we think about it from our knowledge into reaction type, ligases are going to do like combination reactions where we have multiple substrates, but we have a single product. Any questions so far in the definition of an enzyme? What do they do, or the classification of enzymes? Excellent. So as I was mentioning, the active site is the place in the enzyme where we specifically see that substrate binding. And specifically, we can see how the amino acids that are sitting in that active site is going to have specifically interactions. Now, the one that is depicted here only illustrates hydrogen bonds, salt bridges, even hydrophobic interactions between the substrate and the enzyme active site, but understand that these are not the only ones. This is just illustrating that the side chains of the amino acids is what mostly is interacting with the substrate in order for the enzyme to catalyze the reaction that it needs to do on the substrate. Now, when we write an enzyme catalyzed reaction, we're going to actually write it a little bit different from what we have learned of how to write reactions in general chemistry and organic chemistry. Whenever we have an enzyme catalyzed reaction, understand that here we have the components, okay, for our enzyme catalyzed reaction. So when we're writing an enzyme catalyzed reaction, E stands for the enzyme. S is the substrate. And in an enzyme catalyzed reaction, understand that the enzyme and the substrate are going to come together. We can see that then we are going to make what is called an enzyme substrate complex, which is like an intermediate form of the substrate just sitting in the enzyme. Understand that this process is reversible. And the idea is that because the enzyme and the substrate need those different parameters that we learned before in order for a reaction to happen, that includes the right collision, the correct orientation, enough activation energy in order to produce products. All those parameters need to be met in order for the enzyme and the substrate to be committed to making product. So once the enzyme and the substrate are coming together in the right orientation, so the bonds that need to be broken break, that needs to be formed. If we are colliding with the correct energy, if we have enough activation energy, then the enzyme substrate complex turns into just the enzyme and the product. So P is the product that we're going to form. It could be one or it could be multiple. Now, understand that in an enzyme catalyzed reaction, enzymes are regenerated. Enzymes in an enzyme catalyzed reactions are not destroyed, are not linked with the product in order to move forward, okay? What basically happens is that the enzyme and the product are going to separate. The product is released, and now the enzyme has the ability to go back and find a new substrate in order to create that product. So as you can see, this is actually can be visualized like it is a cycle. And in the image that we have here on the right side, we can see the whole process of an enzyme catalyzed reaction. This reaction that we have here is how sucrase comes together with sucrose to make fructose and glucose. So if I write this reaction, it's going to be in the following way. Sucrase plus sucrose forms the sucrase-sucrose complex. And then we're going to form, as a product, the sucrase will be regenerated, and then we're going to have glucose plus fructose. That's basically how we would write it, okay? Now, in this illustration, we can see how the substrate sucrose binds the active site of sucrase, and it forms the enzyme substrate complex. Then, specifically for how sucrase acts on the molecule of sucrose, understand that water comes in as an additional molecule, then the enzyme is able to convert that substrate into the two products, which are glucose and fructose. And then you regenerate that enzyme so it can go back and act on a brand new substrate. So looking at this reaction overall, and since I just mentioned the different classifications of enzymes, since sucrase actually utilizes water to break a bond between glucose and fructose in the sucrose molecule, what will be the classification for a sucrase molecule? So what type of enzyme? Let's put it in the chat. Is sucrase. Great job, Daniela. Yes, this is a hydrolase. Because with water, it cut the substrate into two. Okay? Any questions about how an enzyme catalyzed reaction actually works? Well, enzymes actually have different modes of actions when it encounters a particular substrate. And it has been documented in the literature how different enzymes are going to have different modes of actions. The two well-known modes of actions that are documented for enzymes are the lock and key model, which is what we see on the top, and we also have the induce fit model. So when it comes to the lock and key model, some enzymes understand that their active site is locked in place. The substrate is going to have a particular shape. When they come together to form the enzyme substrate complex, as you guys can observe right here in that middle structure, the substrate and the active site of the enzyme are going to fit into one another, just like a lock and a key. If we utilize that analogy, understand that the key is going to be the substrate and the enzyme is going to be the lock, and the opening where we put the key, that's going to be the active site of your enzyme. So once that substrate is placed in the opening where the key fits in the correct direction with enough energy for overcoming that activation energy, then understand that that enzyme is able to produce product and regenerate that enzyme. So for enzymes that utilize the lock and key model, understand is that that enzyme does not have a lot of conformations in terms of the active site. It's basically, it has the correct space for that substrate, but it doesn't modify itself to accommodate the substrate. Now, other enzymes utilize the induce fit model. And the induced fit model basically means that if you have an enzyme, as you can see in the bottom illustrations for the induced fit model, if you have an enzyme and then you have its substrate, the enzymes that utilize the induced fit model, understand that in this case, the enzyme is going to be a little bit more flexible. It interacts with the substrate because the substrate fits in the active site. But enzymes that utilize the induced fit model are going to adopt conformations, and they're going to basically modify its structure so it's a little bit more flexible, and it's going to accommodate that substrate. So enzymes that utilize the induce fit model are going to be more flexible, okay? Now, once they have adopted the correct conformation, they are going to then break down the substrate into the product and it regenerates. So the main difference between these two models of action for enzyme is just that the lock and key model is less flexible than the induced fit. And understand that it is not generalized that one is better than the other. This is just the models that have been designed just by observation, experimental observation of how the enzymes act on their substrates, okay? Now, enzymes can be affected by a number of factors, okay? And understand that enzyme activity, because remember, they are catalysts. They are going to convert substrates, in other words, reactants to products. So temperature affects enzyme activity, pH affects enzyme activity, and lastly, the presence of inhibitors affects enzyme activity. Now, let's understand that when it comes to the activity of the enzyme, activity specifically is what's going to describe how fast an enzyme catalyzes a reaction from reactant to product. In other words, from substrate to product, okay? Now, let's explore how each one of these three factors, temperature, pH, and the presence of inhibitors, actually affect enzyme activity. The image that you see on the right side of the slide is actually the picture of just hot water. Like, I don't know if anybody has traveled and have seen this water deposits that actually occur in nature, which they are really, really hot waters. Some are too high and you can, oh, there you go. In Yellowstone, there's hot springs. There's some that are too hot and you cannot bathe, but other ones have been designed so people can visit and then just enjoy the natural thermic waters. Now, it is known that there are some of them that have organisms that are known as thermophiles, okay? And what a thermophile is going to be an organism that can survive actually the high temperatures. And those high temperatures, as you can see, can go from 50 degrees Celsius to 120 degrees Celsius, okay, for these hot springs. Now, the interesting thing is that the ability for an organism to be surviving these very extreme high temperature depends on the proteins that are in that particular organism. So understand that these organisms that are known as thermophiles contain a protein profile that allows it to be okay and endure these really hot temperatures. So as you can see, even at the molecular level, we can explain the survival of some organisms, because even though biology will tell us, oh, this is the type of organism that lives there, at the molecular level, those functional groups in the proteins that exist inside of that organism is what actually explains its survival. So now let's explore the three different parameters or the three different conditions that affect protein structure. The first one that we're going to explore is temperature, okay? So how is temperature related to enzyme activity, okay? If we're talking about enzymes that are working on the human body, most enzymes, okay, that we have in the human body, they have an optimum temperature. What is optimum temperature? It's going to be the value of temperature where your enzyme has its maximum activity. That's what optimum temperature is. Let me just write down the definition. Now, you can see it in the first bullet point. What is that optimum temperature for the enzymes in our bodies? Is 37 degrees Celsius, okay? That's why it is so important to have that body temperature in the normal range, because all the biochemical reactions that are undergoing in the body are going to be occurring efficiently because we have the optimum temperature for them to be acting. Okay, and this is just in a normal, healthy human body. So the graph that we have on the right side of the slide, it is just illustrating that that optimum temperature, which is around 37 degrees, is where we have the maximum activity of the enzyme. Now, notice that if we look closely at the blue line, which is going to look at the reaction rate for a particular reaction, it doesn't matter if we go lower in temperature or higher in temperature. The activity of the enzyme is going to be decreasing if it stands away from that optimum temperature. Now, when you have things that are going in this direction, when the body temperature is lowering, notice that the activity is going down, 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 down. Now, why is it that the activity of the enzyme is actually lowering when we're lowering the temperature? There, as you're cooling down the body, then the dynamics and the kinetics of your enzyme, meaning that movement that the molecules need to do, is going to be reduced, okay? So since now the enzyme cannot be moving as fast, basically, since it loses that kinetic energy that it had at its optimum temperature, that's why we see lowering on activity. On the other hand, whenever we are increasing the temperature, we learned before that increasing the temperature actually increases the activity of a particular catalyst, because we know that increasing the temperature creates more collisions and that can increase the reaction rate. Well, this is the thing. Whenever you are increasing the temperature in an enzyme-catalyzed reaction, this is the problem with it. The reason why we see the decrease in activity overall is because as you are increasing the temperature, understand that now the shape of your enzyme is going to be disrupted, because if you increase the temperature of your enzyme catalyzed reaction enough, then your enzyme that is a protein, it can become denatured, okay? So understand that in this process, it is important to note that you have an optimum temperature in order for your enzymes to be active, and deviations either cold or hot will affect enzyme activity and is going to be a little bit different in terms of why is it that when you lower the temperature, there's a lower activity, and then when you increase the temperature, there's going to be a lowering activity. Remember that decreasing the temperature, then we are stopping that kinetic energy. When we increase the temperature of an enzyme catalyzed reaction, pretty much what is occurring is that now we are going towards events that are disrupting enzyme structure, because denaturation is basically what we are going towards. There's a question in the chat. Is this the reason why you want yeast to activate in warm water before adding it to flour? It is one of the reasons, because the thing is, like, if yeast actually have an interesting life cycle. Yeast are very, what is the word that I'm trying to find? Robust when it comes as an organism. For me, I can talk about my experience with yeast. I used to do cancer research when I was doing my PhD. And then when I moved to do my research in Canada, then we started using yeast as a model. Yeast can actually stand different conditions. It can actually survive, you know, at room temperature for days. Okay. If you give it, you know, food, it will survive. When you're working with cancer, you have to have everything regulated, temperature, it needs to be sterile. Using human cells, it's a whole, I don't know, it's it's a little bit complex because there's many things that need to be regulated. Now, whenever you're adding the warm water in order to activate the yeast, basically you are waking them up from that dormant stage. And once they are awake, then for sure they're hungry. So what they go is for the sugar. So it is important, unless you're utilizing fat active yeast, is you need to wake them up. So that's why you use warm water in order to wake them up from that dormant step. I am not much of a baker, so I don't know a lot of the yeast. I only did research with them to understand their biochemical pathways, specifically on how they consume sugars and how they memorize their the sugars that they have consumed, just because it is important in brewing and fermentation. That's basically what I was doing in terms of research. Now, let's move on to the other condition that can affect enzyme activity, which is pH, okay? Now, similar to temperature, pH, enzymes are going to be the most active at a particular pH, okay? So optimum pH is very similar to the definition of optimum temperature. So optimum pH is going to be the pH at which the enzyme has maximum activity. Let me just write that down. Optimum pH is the pH at which an enzyme has its maximum activity. Now, understand that enzymes contain R groups of amino acids, okay, with proper charges at their optimum pH, okay? Let's understand that if you have an enzyme and you know its optimum pH, pH deviations into lower pH or higher pH will affect enzyme activity by reducing it. Now, understand that when an enzyme loses its activity in low or high pHs, okay, is because its tertiary structure is actually disrupted. Why is it its tertiary structure? Because remember that the tertiary structure is where you have the side chains of the amino acids that are going to be mostly experiencing these changes in pH. But one of the important things to note is that when it comes to what is the pH value for an enzyme to be the most active, well, there are different pHs for different enzymes, and it all depends on their location, okay? Because depending on where the enzyme is catalyzing the reactions that needs to catalyze. And let me note to you guys that the human body is such a beautiful creation, because when it comes to looking at the biochemical reactions that are occurring in our bodies right now, the enzymes have a specific substrate. So the design of the human body, you know, whoever you believe it created it, what happens is that we have enzymes that are going to do one job and one job only. We have enzymes that have multiple substrates, but the majority of the enzymes have a particular substrate that they have to act on in order to get to product. That's why we have thousands, tens of thousands of enzymes in our body working on our cells at any given time. Now, when we look at the location of those enzymes, and we can see this illustrated in table 16.11, we can see the different optimum pHs for a selected number of enzymes. And when we compare them, we can observe that depending on the location of the enzyme, then that's where its optimum pH is. So when we look at table 16.11, we can see that, for example, we discussed back in chapter 10 how the stomach is actually going to have gastric juices, and those gastric juices have HCl in it. We know that HCl is a strong acid. So when we look at the enzyme, for example, pepsin, which its location is the stomach, and the reaction that pepsin actually does is that it hydrolyzes peptide bonds, we can see that its optimum pH is also very acidic, between 1.5 and 2.0. So what does this mean? That pepsin is going to have its optimum activity at very low pHs. Now, let's look at, for example, amylase. Amylase is going to be an enzyme that lives in the pancreas. Amylase is an enzyme that actually hydrolyzes amylose, okay? And remember that amylose is one of those two polysaccharides that exist in starch. So when this amylose has been hydrolyzed by amylase, we can see that the optimum pH for that enzyme amylase is between 6.7 and 7.0. So it's from neutral conditions to slightly acidic. Now, if we look at, for example, arginase, which arginase is an enzyme that lives in the liver, and its substrate is arginine because it is in the biochemical pathways of the utilization of arginine. Look at its optimum pH, 9.7. So again, when it comes to optimum pH, we cannot say all enzymes need to be at 7.4, which is the pH of the blood, because that will be incorrect. The correct terminology for optimum pH has to be based on the location of that enzyme in the body. That's where its optimum pH is. Any questions on the conditions of temperature affecting enzyme activity or pH affecting enzyme activity? Then let's move on to the last condition that can affect enzyme activity, and that is the use of inhibitors. Understand that inhibitors have been utilized in the world of medicine in order to treat specific conditions or manage specific conditions. That's why inhibitors are important in medicine. Now, there are different classes of inhibitors. In this course, we only compare 3 different inhibitors: a competitive inhibitor, a non-competitive inhibitor, and an irreversible inhibitor. Those are the only 3 that we're going to learn. So by definition, a competitive inhibitor is going to be, and it's all summarized in table 16.12. A competitive inhibitor is going to be an inhibitor that when you compare the shape of the substrate to the shape of the inhibitor, they are going to be similar, okay? So again, in a competitive inhibition system, the shape of the substrate and the shape of the inhibitor is going to be similar. They are not going to be exact the same because they're going to be identical molecules, but they're going to be very similar, that they may be like a few functional groups away from each other. Now, when it comes to, now that you know that the inhibitor has, a similar shape to the substrate, how does it bind the enzyme in order to inhibit it? And inhibition basically means that it's going to affect the activity of the enzyme by not allowing it to be active or by reducing its activity. Well, in a competitive inhibitor system, then that inhibitor is going to compete with the regular substrate to sit in that active site, okay? Now, competitive inhibitors, they are actually reversible. The reason why they are reversible is that you are not affecting that active site being attracted to its regular substrate. So if you add more substrate, you can basically reverse having that inhibitor present in your enzymatic reaction. Understand that in the research, which I have utilized inhibitors just to study protein function, they are very useful because the use of inhibitors in research is going to allow us to just, put some enzymes in states in which they're not now in the whole picture that you're trying to study in research. So then you can see how the system is going to interact with each other without having the enzyme there. And it depends on what you're trying to study. This is a very important insight into how biochemical pathways actually work. So we can see an illustration of how a competitive inhibitor acts. If you look at letter A, that illustrates the substrate, the active site, and the enzyme. You can see in letter B that your competitive inhibitor is going to sit in the active site, and now the substrate cannot bind the enzyme. Now, when it does this, understand that in competitive inhibitors, we are going to look at interactions that are just happening in the active site. So the inhibitor really doesn't bind chemically, meaning it doesn't form a covalent bond to the active site. That's why if you add more of the natural substrate, you can compete it out. So let's move on to the other type of inhibitor, which is non-competitive inhibitor. And when we look at table 6.12, a non-competitive inhibitor does not look like the substrate, okay? The non-competitive inhibitor, actually its role in the whole definition of how it inhibits an enzyme is that it's going to bind in a site in the enzyme that is not in the active site, okay? So when a non-competitive inhibitor binds the enzyme, because this site is away from the active site, that is known as an allosteric site. That's how we define it. So allosteric means that is binding to a site in the enzyme that is not the active site. Now, understand that this molecule binding at the allosteric site for the enzyme actually affects the shape of the active site, and that's where the inhibition comes from. Remember that I explained a few moments to you guys that, or a few moments ago to you guys, that when it comes to substrate binding to that enzyme, it needs to do it in the correct shape, orientation, with enough energy to overcome the activation energy. And now if you change the shape of the active site by the non-competitive inhibitor, even if the inhibitor is present, then it's not going to have the ability to bind the enzyme because it doesn't have the right shape anymore. Now, it is important to note that when it comes to non-competitive inhibitors, if you add more substrate, that is not going to reverse the action of the non-competitive inhibitor. What you need to do in order to reverse a non-competitive inhibitor is that you have to chemically change the enzyme to remove that inhibitor. That's what we know to date. Lastly, we have irreversible inhibitors. Now, similar to non-competitive inhibitors, when we look at the shape of the inhibitor to the substrate, they do not look the same, okay? So that's how irreversible inhibitors are, in a way, I guess, by definition, just similar to non-competitive inhibitors. The thing about what makes a irreversible inhibitor have its name is that it forms covalent bonds with the enzyme, and that's how it acts. So if it's an irreversible inhibitor, then understand that that covalent bond is permanent and we cannot reverse it. The classical example that we always talk about when it comes to irreversible inhibitors is the use of penicillin. When penicillin was discovered and used, understand that this changed the world of medicine forever. Because during that time when penicillin was not available, people were dying of infections. So once penicillin was discovered, and it was observed that penicillin actually attached to, when you have bacteria, we didn't talk about their shape, but understand that penicillin actually gets into the bacterial cell and it starts preventing biochemical reactions from happening, specifically the outer core of the bacteria. I don't want to talk about in terms of a lot of details because we didn't talk about how bacteria look like, but understand that when it came to penicillin, then once people started getting treated by penicillin, then people were being cured from those infections. Now, the other hand of use of penicillin is that some people have developed allergies to penicillins. So new molecules or derivatives of penicillin have to be discovered and used for treatments. And also another thing is that, as you, for those of you that have taken micro, you may have heard that sometimes these bacterial species are going to create modifications inside of their biomolecules and create resistance to these antibiotics. And for those of you that work in hospitals or have heard of these situations, right now we have in this world different organisms, specifically bacteria, that are resistant to these treatments of antibiotics. So it is a problem right now. So the overuse of antibiotics has also created a problem in modern medicine, okay? Any questions about inhibitors? Yeah, MRSA. The multiple resistance. It is incredible. Like it is, for me as a biochemist, it's very interesting to see how organisms adapt. And now that in chapter 17, we're going to talk about DNA and all that stuff, the rate at which these organisms, adapt and modify their genetic material in order to just resist this chemical treatments that we try to give them to kill them. It is interesting. Again, if you have taken microbiology, maybe this is one of the topics, because I'm not aware of it. I never taken micro. I took bacteriology when I was in undergrad. But even then, understanding these processes, it is something that as researchers, we continue to explore, just because in everyday life, we see these organisms still being around us, and then we have to create solutions in order to deal with them. Any questions on chapter 16? So this is the end of chapter 16. And now let's move on to the last chapter of material that we have for the incoming exam, which in chapter 17, we are now going to talk about nucleic acids. Then we are going to proceed to talk about protein synthesis. So throughout our conversations in biochemistry, you guys have been able to observe 3 out of the 4 biomolecules that are important in the body. In chapter 13, we talked about carbohydrates. Unfortunately, in this class, this summer semester, we're not going to talk about how carbohydrates are going to turn into energy. Then in chapter 15, we talked about lipids, which we observed how lipids are very important in terms of the structures of the cell, but is also a biomolecule that we store in our body for long-term energy. We store carbohydrates, but remember, carbohydrates is for short-term energy. We just finished talking about amino acids and proteins. Amino acids and proteins overall, they are going to carry many, many functions in cells, but understand that the amino acids and proteins that we have in the body, we are not storing them for energy. It's only under extreme cases in which if somebody is suffering from severe starvation, that then the body triggers, okay, processes in order to derive energy from the proteins that we have in the body. But normally, when you look at the human body, we don't have a storage. We don't have, like, for example, a molecule like glycogen that we know is used for energy, or for, in the case of lipids, triglycerides, we know that that is for energy. We do not have, yeah, that's a great point, Gia, exactly. So muscle atrophy, which you can see it in people that are undergoing extreme starvation. Like at that point, your body is really scavenging for anything that can utilize for energy. But normally, the body does not have a single molecule that we say, oh, this is how we store proteins. We don't do that. We don't do that. Like the body doesn't store proteins for later use. It has to be under extreme conditions that then the body goes for the proteins and muscle in order to create energy. Again, it's all, we do not store protein for energy is what I'm trying to say. Now, in chapter 17, we are going to talk about the last biomolecule that it is important in the human body in order for the body to communicate what is needed in the cell depending on environmental cues. Okay, so we are going to discuss what are nucleic acids and the whole process of protein synthesis. So I already mentioned to you guys that it is important to understand the different compartments of an animal cell. And when it comes to nucleic acids, understand that nucleic acids live in the nucleus, okay? And they're synthesized and, replicated in the nucleus. One of those nucleic acids that are important, and it's basically the basis of synthesizing other biomolecules, is DNA. And if you have taken biology classes, you have heard that, oh, DNA turns into RNA, RNA turns into protein. And yes, that is the central dogma of how genetic information is passed on to a cell. This happens in different compartments. So understand that when we are talking about these processes for nucleic acids, we're going to focus on the nucleus. We're going to focus on the cytoplasm. And we're also going to focus on the ribosome. The reason is that in the nucleus, this is going to be the compartment of the cell where we have storage of one of the nucleic acids, but we are going to see how making these nucleic acids also happen in the nucleus. In the cytoplasm is where we are going to be transporting some of these nucleic acids in order to make proteins, okay? And lastly, when it comes to the ribosome is going to be those little dots that you see in the cell. And in the ribosome, that's where we have the machinery inside of cells in order to make protein, okay? So let's get started understanding nucleic acids overall. So what is a nucleic acid? Okay. A nucleic acid is going to be a polymer of nucleotides. Okay. So let me just write the definition. A nucleic acid is going to be a polymer of nucleotides. Now, this means that nucleotides are going to be the basic unit for a nucleic acid. In the body, we have two important nucleic acids. We have DNA and we have RNA. So when we look at them in terms of their definition, understand that DNA is going to be a polymer of deoxyribose nucleotides. RNA is a polymer of ribose nucleotides. Okay. Now, because this is a chemistry class, and for those of you that have taken biology, these concepts may not be foreign to you, but we are going to understand this DNA and RNA at the chemical level. That's what I'm bringing to the plate. You may know about DNA, and then RNA is made from DNA and where all of that happens. But in this class, now we're going to focus into observing, okay, these important polymers, and even at the bases, their nucleotides in terms of their structures. Now, we know that since nucleotides are going to be specifically the basic unit for a nucleic acid, we can observe that when it comes to a nucleotide, nucleotides are made by a base, a sugar, which it can be deoxyribose or ribose. So we have a base, we have a sugar, and phosphate. So if you have those three components, base, deoxyribose, and phosphate, that is known as a nucleotide. If you only have the first two components, which is base and sugar, then understand that what you have is a nucleoside. So the nucleoside is going to be the version without the phosphate. So before we start incorporating the phosphates, that's what we know as a nucleoside. But understand that for nucleic acids, nucleotides is what we utilize. And you can see here in table 17.1 that we also have the list of the sugars. Now, what you guys can observe here when it comes to the bases is not that we have sodium hydroxide, potassium hydroxide, ammonia. No. The bases in nucleic acids are actually called nitrogenous bases. And the reason why they're called nitrogenous bases or nitrogen-based bases is because in these bases, what we have is heterocyclic amines. To illustrate, okay, this nitrogenous bases, see in the bottom of here, we have the nitrogenous bases, specifically adenine, okay? So this portion of the molecule, this is the nitrogenous base, or the nitrogen base, adenine. And when we look at the structure of adenine, we can see that adenine is a heterocyclic amine. Remember, a heterocyclic amine is going to be a cyclic structure in which now you have nitrogen as an outside atom other than carbon. So that's why they're nitrogenous bases or heterocyclic amine. Nitrogen is in the cycle. Now, let's understand that the linkage for each of these components, okay, in a nucleoside or a nucleotide, it is important to note. So again, when we look at the molecules, and now I'm going to focus on the nucleotide, just because that is the basic unit for a nucleic acid, you can see that there's different positions in it, okay? Now, if I number the carbon atoms, which we only see the fifth one here, this is carbon 1, this is carbon 2, this is carbon 3, this is carbon 5. So it doesn't matter if we have ribose or deoxyribose. What will be the classification of ribose or deoxyribose based on the number of carbons? Let's review that about chapter 13. So a ribose or a deoxyribose, what is going to be its classification depending on the number of carbons? Excellent job, Gia. Yes, they are pentoses. Okay. Oh, sorry, I put a 5. Let me just change this to a 4. So there's 5 carbon atoms. Now, let's understand that the way that these components are bonded to one another is very specifically. As you guys can see illustrated in the nucleoside, carbon number 1 is going to be bonded to my sugar motif through what is called an N-glycosidic bond. And it is called an N-glycosidic bond because a nitrogen in my nitrogen base is bonded to the sugar. That's where the word glycosidic comes from, okay? Now, we can see just by looking at the structure that the sugar is bonded to the phosphate. Where specifically? At carbon 5 in the sugar, that's where we have the phosphate group. Now, the nucleotides, understand that this is the one that we have illustrated here. There's only one phosphate, but understand that in the cell, we can have nucleotides with 2 phosphates or 3 phosphates. And ATP, which is a molecule that I keep on mentioning to you guys, which is utilized in order to make the polymers of nucleic acids and is also utilized for energy in the cell, is going to be this structure that we have here. But instead of with 1 phosphate, is going to have 3 phosphate. That's why the T will come from in ATP. It is adenosine 5' triphosphate. That's what ATP is, okay? Now, let's look at least the nitrogen bases and the sugars for each of the respective nucleotides. So what happens with these nitrogen bases or nitrogenous bases, as I mentioned to you guys, is that they are going to be heterocyclic amines. Now, when it comes to the classifications of the nitrogenous bases that we see in biological systems, some are going to be pyridines, some are going to be purines, okay? The pyrimidines, you can see on the top, we have cytosine, thymine, and uracil. So out of those three, understand that thymine is only found in DNA, uracil is only found in RNA, okay? Cytosine out of your pyrimides is actually found on both DNA and RNA. Now, when it comes to the purines, and you can see the structures of the nitrogen bases that are purines, we have adenine, which we just talked about, and guanine. Both adenine and guanine can be found in the nucleic acid polymers for DNA and RNA. And you can look at the difference between pyrimidines and purines, specifically by looking at the structures in the bottom left corner of the slide. When it comes to these nitrogen bases, they have a one-letter code. And as you can see here, adenine, it is symbolized by the capital A, guanine, capital G, cytosine, capital C, thymine, capital T, and then uracil, capital U. That's going to be the difference between them. Now, how do you know which sugar is present in a particular nucleic acid? The name already told us that in DNA, we have a deoxyribose. In RNA, we have a ribose. So the difference between the deoxyribose and ribose is specifically at carbon number 2. As you can see, the difference between ribose and deoxyribose is that deoxy means that it doesn't have an OH atom at carbon 2. So remember, overall, we saw that carbon 1 in my sugar is what bonds to the nitrogen base. Carbon 5 through its OH binds to the phosphate. Carbon 2 is the one that tells me if I have a ribose or deoxyribose. Another important position in our sugars is going to be position 3, but that we're going to study a little bit later in this chapter. Any questions so far on the definitions of the different nucleic acids or their components? Since there are no questions, right now we're going on break.